While you're reading nutrition labels, trying to avoid high-fructose corn syrup, and wondering why your whole-grain cornbread leaves you hungry an hour later, consider this. The corn you're eating is nutritionally locked. The niacin is chemically bound. The essential amino acids are missing. The mycotoxins are active. You're consuming a grain that, without a specific chemical process involving wood, ash, and water, cannot sustain human life. Indigenous Americans knew this 3,000 years ago. They engineered both the plant and the chemistry to unlock it. Modern industrial agriculture took the seed but abandoned the science. The result is a crop that fuels epidemics instead of empires. Maize does not exist in the wild. You will not find it growing in any forest, prairie, or riverbank on Earth. It is a human invention, the product of a multi-generational genetic engineering project that began approximately 9,000 years ago in the Balsas River Valley of Mexico. The raw material was teosinte, a wild grass that produced seeds encased in a mineralized fruit case hard enough to crack a human tooth. This stone-like gloom was engineered by evolution to survive passage through animal digestive tracts, ensuring long-distance seed dispersal. For humans, it was an impenetrable barrier. The plant itself was bushy, with many lateral branches called tillers, each producing tiny seed-poor ears. When the seeds matured, the ear shattered, exploding to scatter seeds across the landscape for propagation. This shattering mechanism made harvest nearly impossible. Seeds launched themselves into the surrounding vegetation before collection. Indigenous agronomists had to invert nature's entire design. They needed a plant that concentrated energy into a single stalk, created accessible kernels, and retained seeds on the cob for harvest. They achieved this through targeted selection for two critical genetic mutations over countless generations. The first was the TGA1 gene, teosinte gloom architecture 1. This master regulator gene controls the hardness of the seed casing. By selecting plants with a mutation in TGA one generation after generation, indigenous engineers gradually softened the rock-hard gloom into the papery chaff now trapped inside the corn cob. The kernel became exposed and edible. This was not a single discovery, but a sustained program of observation and selection spanning centuries. The second modification targeted the TB1 gene, teosinte branched 1. This gene suppresses the growth of lateral branches. By selecting for high TB1 expression, the plant was forced to redirect all photosynthetic energy away from many small branches and into a single, massive apical stalk and one large female inflorescence, the ear. Instead of a dozen tiny seed heads, the plant produced one enormous cob. The result was a biological paradox. Maize became incapable of reproduction without human intervention. The kernels are wrapped tightly in husks that prevent dispersal. If an ear falls to the ground unharvested, the seeds germinate in a dense cluster where they compete for resources and die within weeks. The parent plant chokes its own offspring. Maize requires human hands to shuck, shell, and plant it. It is a companion species engineered for total mutual reliance. The plant cannot survive without us. We engineered it that way deliberately. This was deliberate bioengineering, not accidental domestication. Maize is the world's first genetically modified organism, and it was developed without the use of laboratories. The ancestral corn varieties used across colder climates were flint corns, named for their physical structure. Unlike modern yellow dent corn, which has a soft, chalky starch center that degrades easily, flint corn consists of a tiny, soft core surrounded by a thick layer of hard, glass-like vitreous starch. The kernel resembles a geological formation more than a grain. This hard shell is hydrophobic and airtight, creating a hermetic seal. Flint corn resists mold spores, weevil penetration, and moisture damage. The kernel acts as its own preservation chamber. This was critical engineering for winter survival in northern climates. Indigenous communities stored flint corn in underground caches, earthen silos lined with bark or grass. The grain remained viable for multiple years without spoilage. There were no propane dryers, no chemical fumigants, no refrigeration units, no controlled atmospheres. The material science of the kernel itself provided the preservation technology. The architecture of the grain made long-term storage possible. Modern industrial corn, by contrast, requires immediate artificial drying after harvest. Yellow dent's soft starch center acts like a sponge, absorbing atmospheric moisture. The grain begins rotting within weeks if left untreated. Modern corn requires massive propane-fueled dryers that blow heated air through grain bins for days, substantial energy cost, and absolute infrastructure requirement. Without industrial drying systems and chemical treatments, yellow dent degrades rapidly. The primitive grain was engineered to outlast the modern one without any external inputs whatsoever. The same physics that made flint corn shelf-stable also made it the ancestor of popcorn. Water trapped inside the hard shell cannot escape during heating. Internal pressure builds until the starch gelatinizes and the kernel explodes, turning inside out. The hull acts as a natural pressure vessel. The explosion is not a cooking accident. It is engineered physics. Storage capacity determined survival. A community that could preserve grain for 2 or 3 years could endure consecutive failed harvests, catastrophic weather events, or pest invasions that destroyed a single season's crop. Flint corn's architecture was not incidental. It was engineered survivability translated into seed structure. Maize is a heavy nitrogen feeder. It strips nitrates from the soil rapidly as it grows, depleting the primary nutrient required for chlorophyll production and vegetative growth. Modern industrial agriculture compensates with synthetic anhydrous ammonia fertilizers, which require massive fossil fuel inputs for production and distribution. Indigenous engineers solved nitrogen depletion by interplanting beans. Legumes possess specialized bacteria called rhizobia in their root nodules. These bacteria exist in a symbiotic relationship with the plant, receiving sugars from photosynthesis while performing nitrogen fixation. The bacteria capture atmospheric nitrogen, N2, and convert it into ammonia, NH3, a form of solid nitrogen fertilizer that is deposited directly into the surrounding soil during the growing season. This is biological nitrogen fixation operating in real time. The beans actively refuel the soil while the corn is still growing, eliminating the need for crop rotation or fallow periods. The corn extracts nitrogen. The beans replace it. The system is regenerative rather than extractive. But the engineering is multi-layered beyond nitrogen dynamics. The corn stalk simultaneously functions as a vertical trellis for the climbing bean vines. This eliminates the material cost and labor of constructing separate support poles from wood or installing wire frameworks. The beans, in turn, wrap around the corn stalks in a helical pattern, anchoring them against high winds. This reduces lodging, the term for when tall plants fall over during storms. The bean vines act as guy wires, distributing wind load across the structure. The third component is squash, planted at the base of the corn and bean stalks. Squash produces massive, broad leaves that can span 2 feet in diameter. These leaves function as living mulch, creating a microclimate at ground level. The shade lowers soil surface temperature, drastically reducing evaporation rates. The soil remains moist, longer between rainfall events. The darkness under the squash canopy also prevents photosynthesis for competing weeds. Annual weeds require sunlight to germinate and establish. The squash leaves block that light completely, eliminating the need for herbicides or hand weeding. The squash vines themselves are covered in stiff spines and rough trichomes, creating a physical deterrent that prevents raccoons and deer from entering the plot to feed on the ripening corn ears. The Three Sisters system achieves nitrogen fixation, structural support, water conservation, and pest defense through species interaction alone. The system is self-regulating because it was designed around ecological physics rather than fighting them with chemistry. Maize contains vitamin B3, also known as niacin. The vitamin is present in substantial quantities within the kernel. But the niacin in raw corn is chemically bound to complex carbohydrates and peptides in a compound called niacin. The human digestive system cannot break these ester bonds. The enzymes in our saliva, stomach acid, and intestinal tract lack the chemical capacity to cleave the bonds holding the vitamin to the carbohydrate matrix. The vitamin is present but biologically unavailable. It passes through the digestive system without being absorbed. Raw maize is also deficient in two essential amino acids, lysine and tryptophan. These amino acids are called essential because the human body cannot synthesize them internally. They must be obtained from food. Lysine is required for protein synthesis, calcium absorption, and collagen formation. Tryptophan is the precursor for serotonin and melatonin, neurotransmitters that regulate mood, sleep, and cognitive function. Without adequate lysine and tryptophan, the body cannot build muscle tissue, repair damage, or maintain neurological function. This means that eating raw corn provides starch, energy in the form of glucose, but lacks the molecular building blocks necessary to sustain human physiology over time. It is a carbohydrate source that delivers calories, but not the amino acids required to convert those calories into functional tissue. You can eat raw corn until your stomach is full and still be starving at the cellular level. This is not a hypothetical problem confined to historical populations. It is the chemical reality behind modern whole-grain cornmeal, corn chips, tortilla chips labeled as natural, and cornbread marketed as nutritious. If the corn has not been treated with an alkaline solution before milling, the niacin remains locked, the amino acids remain deficient, and the mycotoxins, if present from storage mold contamination, remain active and carcinogenic. The grain provides empty calories in the truest biochemical sense. Energy without structural nutrition. This is why populations that adopted corn as a staple without understanding the required chemical processing developed widespread deficiency diseases that devastated entire regions. The problem was not the crop itself. The problem was consuming it without the chemistry that transformed it from bulk starch into food capable of sustaining life. The solution was nixtamalization, a chemical transformation that alters the molecular structure of the grain, not a cooking method. Indigenous chemists boiled dry corn in water mixed with wood ash, which contains potassium hydroxide, or burnt limestone or river clam shells, which contain calcium hydroxide. These are highly alkaline substances with a pH between 11 and 13, caustic enough to burn skin. The corn was boiled in this solution for a period ranging from 20 minutes to several hours, depending on the variety and hardness of the kernel, then left to steep overnight in the cooling liquid. The following day, it was rinsed thoroughly with clean water to remove the pericarp, the outer hull, which had been loosened by the chemical treatment. Multiple chemical reactions occurred during this process, operating simultaneously. The alkaline solution degraded the hemicellulose, a complex polysaccharide that glued the hull to the kernel interior. This chemical degradation skinned the corn and made it grindable into a smooth dough called masa. The hull separated cleanly, leaving the starchy endosperm exposed. The high pH also hydrolyzed the ester bonds that locked the niacin to the carbohydrate matrix. Hydrolysis is the breaking of chemical bonds through the addition of water molecules in a high pH environment. This reaction released the vitamin into a free, bioavailable form that human enzymes can recognize and absorb. Niacin availability increased from nearly 0% to 100% through this single chemical step. The process altered the protein structure. While total protein decreased slightly, the availability of lysine and tryptophan increased by up to 2.8 times as the alkaline treatment broke down structures that sequestered these amino acids. This shift brought the amino acid profile closer to completeness, especially when the nixtamalized corn was eaten with beans, which provided complementary amino acids. The corn absorbed calcium during the overnight soak, increasing calcium content by 750%. In a culture without domesticated dairy animals, nixtamalized corn provided the bone density mineral essential for skeletal health, dental integrity, and cellular signaling. This was the calcium source that enabled the construction of healthy bones in populations that never consumed milk. The alkaline bath destroyed 97 to 100% of aflatoxins, potent carcinogens produced by storage mold. The high pH denatured these toxins, rendering them harmless. The chemical treatment neutralized a cancer threat that modern grain storage still struggles to control. Nixtamalization converted a locked carbohydrate into complete protein, fortified it with calcium comparable to dairy, and removed fungal carcinogens. This was molecular engineering applied with precision. When Christopher Columbus and later Spanish explorers encountered maize in the Americas, they recognized it as a high-yield staple crop capable of supporting large populations. They transported the seeds back to Europe, where corn was rapidly adopted across Spain, Italy, Romania, and parts of Africa as an affordable food source for the growing peasant and urban poor populations. But they did not transport the chemistry. European observers witnessed indigenous people soaking corn in ash water and grinding it into dough, but they interpreted this as a flavor enhancement technique or a cultural ritual without functional purpose. They did not understand it as an essential biochemical process required to make the grain nutritionally viable. They saw a ceremony where there was science. Europe began cultivating corn and consuming it raw, milled into polenta, grits, and cornmeal without alkaline treatment. For a time, this seemed adequate. Corn provided cheap calories for rapidly growing populations during a period of demographic expansion. The grain produced more calories per acre than wheat or barley. It seemed like an agricultural miracle. Then people began to sicken. The disease was called pellagra, from the Italian pelle agra, meaning rough skin. It was characterized by 4 symptoms, known grimly as the 4 Ds: dermatitis, diarrhea, dementia, and death. The skin lesions appeared first as dark, scaly patches on sun-exposed areas. Digestive failure followed, then neurological deterioration. Without intervention, the disease was fatal. By the early 20th century, pellagra had become epidemic across multiple continents. In the American South alone, where poor sharecroppers subsisted on cornmeal and little else following the collapse of the plantation economy, 3 million people were infected. 100,000 Americans died. The medical establishment initially believed pellagra was infectious, possibly caused by a pathogen in spoiled corn. It was not until 1914 that Dr. Joseph Goldberger, a U.S. Public Health Service physician, conducted experiments proving that pellagra was a nutritional deficiency disease caused by a diet overly dependent on untreated corn. Goldberger demonstrated that the disease could be prevented and cured by consuming niacin-rich foods like meat, eggs, and milk, or by treating corn with alkali before consumption. Goldberger had rediscovered through controlled scientific experimentation what indigenous Americans had known and practiced for 3,000 years. The pellagra epidemic was not caused by ignorance of an unknown fact. It was caused by arrogance, the assumption that indigenous food processing was primitive superstition rather than advanced chemistry developed through generations of observation and refinement. The cost of that dismissal was measured in hundreds of thousands of preventable deaths. A traditional indigenous cornfield contained genetic diversity visible to the naked eye. A single ear might display kernels in red, blue, black, white, and yellow, each color representing a different genetic lineage. This was risk management encoded into agricultural practice. Different colored kernels represented different genetic varieties with different environmental tolerances. Blue corn thrived in wet seasons but rotted in waterlogged soil. Red varieties survived drought with deeper roots but struggled in flooding. White corn performed well under moderate conditions but poorly under extremes. By planting all varieties together, the farmer created a genetic portfolio. If the season turned wet, blue corn produced strong yields while red corn failed. If drought struck, red survived while blue withered. The crop was resilient because it was diverse. Failure was distributed across genetic lines rather than concentrated in a single vulnerable genotype. The same principle applied to disease resistance. A pathogen that devastated blue corn left red and white untouched. Total crop failure was nearly impossible because no single threat could exploit every genetic variant simultaneously. Modern industrial agriculture operates on the opposite principle. Fields are planted with genetically identical F1 hybrids, bred for uniform maturation, height, and kernel moisture. Every plant in a 1,000-acre field shares the same genome. This uniformity enables mechanized harvesting. Machines require predictability. The entire field must mature simultaneously for a single pass harvest, but it creates systemic fragility. If a new pathogen emerges that exploits a specific genetic vulnerability, every plant in the field is susceptible. There is no genetic firebreak, the entire crop can be lost. This is not theoretical speculation. In 1970, southern corn leaf blight, a fungal disease caused by Bipolaris maedis race T, swept through US cornfields. The epidemic destroyed 15% of the national corn crop in a single season because the majority of hybrid corn varieties planted that year shared a genetic trait called Texas male sterile cytoplasm, which made them uniformly vulnerable to the pathogen. The fungus found a monoculture and burned through it like wildfire through dry grass. The epidemic revealed the cost of monoculture. Harvest efficiency had been purchased at the price of biological resilience. Indigenous farmers had solved this a millennium earlier. Diversity was infrastructure against total failure. Modern yellow dent corn was not bred for human consumption. It was bred for maximum yield per acre, measured in bushels. Nutritional density was not a selection criterion. Compared to heirloom Flint varieties, yellow dent corn contains significantly lower concentrations of protein, calcium, zinc, and iron. The primary uses of modern field corn reflect this reality. 36% of the crop becomes animal feed for cattle, pigs, and chickens in confined feeding operations. 40% is converted into ethanol fuel, an additive for gasoline that provides modest octane enhancement at substantial environmental cost. The remainder is processed into industrial derivatives, the most ubiquitous of which is high-fructose corn syrup. HFCS is produced through acid enzyme hydrolysis. Corn starch is dissolved in a solution of hydrochloric acid, then treated with industrial enzymes, alpha amylase and glucoamylase, that break the starch chains into individual glucose molecules. A final enzyme called glucose isomerase converts a portion of the glucose into fructose, creating a syrup that is 55% fructose and 45% glucose. The result is a liquid sweetener that is cheaper to produce than cane sugar, easier to transport in tanker trucks, and simpler to blend into processed foods. It is now present in soft drinks, fruit-flavored beverages, baked goods, breakfast cereals, condiments, salad dressings, and processed foods across the industrial food system. The average American consumes roughly 60 pounds of high-fructose corn syrup per year. But fructose and glucose are metabolized by fundamentally different biochemical pathways. Glucose is processed by every cell in the body and triggers insulin and leptin, hormones that regulate blood sugar and signal satiety. Fructose is metabolized almost exclusively by the liver, bypasses the insulin response, and does not trigger leptin. When the liver is flooded with fructose beyond its oxidative capacity, it converts the excess directly into fat through a metabolic pathway called de novo lipogenesis. The liver synthesizes triglycerides, fat molecules, from the fructose and stores them in its own tissue. These fats accumulate in hepatocytes, leading to non-alcoholic fatty liver disease, a condition previously rare outside of chronic alcoholics, but now epidemic in populations with high HFCS consumption. Pediatric liver disease, once almost unheard of, is now common. Fructose also does not trigger the hormonal signals that regulate appetite. The body does not recognize liquid fructose as food in the metabolic sense that it recognizes solid carbohydrates. People continue consuming HFCS sweetened beverages without feeling satiated, leading to chronic caloric overconsumption. Insulin resistance develops. Type 2 diabetes follows. Nixtamalized corn combined with beans produced glucose that fueled cellular respiration and complete amino acids that built muscle and tissue. High fructose corn syrup produces triglycerides that accumulate as visceral and hepatic fat, insulin resistance that precedes diabetes, and metabolic syndrome that shortens lifespan. The plant is the same species, but the processing determines whether it sustains life or damages it. One path was designed by indigenous chemists to unlock nutrition. The other was designed by industrial food engineers to maximize profit margins and shelf stability. Maize was engineered over thousands of years to be a symbiotic partner, a plant that could not survive without humans and humans that thrived because of it. The genetic modifications that created it, the storage physics that preserved it, the nitrogen-fixing polyculture that sustained it, and the alkaline chemistry that unlocked it represent an integrated system of biological and chemical engineering that required no external inputs beyond human knowledge and labor. Modern industrial corn inverted every principle that made the original system functional. It was bred for yield, not nutrition. It is grown in monoculture, not polyculture. It depletes soil instead of regenerating it. It requires synthetic fertilizers derived from fossil fuels instead of biological nitrogen fixation. It is processed into a metabolic toxin instead of a complete food. The difference is not the plant. The difference is whether the system was designed to sustain human life or maximize profit margins within. Quarterly earnings reports. Indigenous Americans built the former over millennia. Industrial agriculture built the latter over decades. The chemistry still works. Nixtamalization still releases niacin, fortifies calcium, and destroys mycotoxins. The Three Sisters still fix nitrogen, conserve water, and suppress weeds. The genetic diversity of heirloom varieties still provides adaptive resilience against climate variance and pest pressure. The knowledge was never lost, it was dismissed, it was categorized as a primitive ritual rather than recognized as an advanced science. And the cost of that dismissal is measurable in deficiency diseases that killed hundreds of thousands, metabolic epidemics that now affect millions, and degraded soil that requires ever-increasing chemical inputs to produce ever-decreasing nutritional value. The primitive crop was a biological masterpiece. The modern one is a profitable mistake.